Bismillahir Rahmanir Rahim. Dear students, Assalamu alaikum and welcome back to our cell biology, genetics and evolution class. After finishing the topic of transcription, now we are going to start translation so as to complete the topic of gene expression. As we saw in the early videos, the gene expression basically consists of, if we keep it short and simple, two basic stages: transcription and translation. So we discussed transcription in the previous videos, and now we are going to start the process of translation and see how it takes place. So protein synthesis, translation, and that is the genetic code which is contained within the messenger RNA. It will be translated into a polypeptide chain. Now this is a general diagram telling you or showing you the overall process of translation. This is a prokaryotic cell. From DNA we have obtained a messenger RNA through the process of transcription, and now the translation is going on. So this big block which you see brown color, this is the ribosome. This one is the large subunit and this one is the small subunit. This particular band which is passing through in this direction through the ribosome between the large subunit and the small subunit is the messenger RNA. And these are blue shaped structures are the transfer RNAs carrying their amino acids to this translation site, where these amino acids are bonded to one another, and you see this particular growing polypeptide chain which is being synthesized. Now these transfer RNAs, they carry their amino acid, bring it here to the translation site, and those amino acids are bonded by the ribosomes, as we shall see in the next videos. So this is the overall process of translation. This particular video, this particular diagram is showing how translation, or what happens during translation. Now the translation process requires the components, and we are aware of most of those components. So transfer RNA, the ribosomes and the messenger RNA. Our discussion of translation process or the protein synthesis, it will be quite simple and mostly restricted to prokaryotes, and you shall study in detail the translation process inshallah in the coming semesters. So these are the three major components: transfer RNA, ribosomes and messenger RNA. So we shall take a look at them one by one, except mRNA, because we have just seen how messenger RNA is synthesized, and in eukaryotes how it is modified and processed. So we are going to take a look at transfer RNA and ribosomes in a very simple way, and then we shall proceed to the start of protein synthesis, actual synthesis of polypeptide chain, or what we call translation of messenger RNA. So the structure and function of transfer RNA, each transfer RNA, what it does, that it translates a given mRNA codon into certain amino acids. So as we shall see in the next slide, the tRNA is basically a translator, what they call an interpreter. Interpreter is a person who can translate one language into another. So this is what transfer RNA does, that it translates the language of the amino acid, that is the messenger RNA, into language of transfer RNA translates the language of nucleic acids, which is basically nitrogenous bases or codons, into the language of proteins, which is amino acids. So this is what it does. Now a transfer RNA, one thing that you must have in mind that a transfer RNA must carry a specific amino acid. A transfer RNA cannot carry different amino acids. It will carry only one, and it will always carry that particular amino acid. For example, if a transfer RNA is made for glycine, then it will always carry glycine and it will not carry other amino acid. So in a very simple way, a transfer RNA has at its one end its designated amino acid, say it is glycine, GLY. And at the other end, it has the three bases which are opposite to the three bases in the messenger RNA. So we know that the three triplets, three bases in the messenger RNA, or the words in the messenger RNA, are known as codons. So the messenger, this is a tRNA. Now this tRNA is carrying at one end an amino acid which is specific to it, and at the other end it has three bases which are opposite to the codon. So these three bases are opposite to the codon. Now glycine has four codons which start with the GG. So GGU and GGA, GGC, GGC and triple G. So we can write any one of them, but that will be opposite to these codons. So let's say it is this one. So it will be CCA. So this is the anticodon because it is carrying glycine. So this CCA will attach to this particular codon in the messenger RNA. So a transfer RNA bears a specific amino acid at one end, while at the other end is a nucleotide triplet, and that can only base pair with the complementary codon. So it is known as anticodon. So at other end it is an anticodon. Now transfer RNA molecules are not very big one. Each transfer RNA is a small molecule about 80 nucleotides long. So the transfer RNA are adapter type of molecule, small molecules, and since it carries many complementary pieces of nucleotides in its sequence, so they can bond to one another. So due to those complementary pieces or stretches of nucleotide bases, it can fold back onto itself and assume a three-dimensional structure, as we shall see in the next diagram. So these are three diagrams of transfer RNA. Now this is a two-dimensional diagram. So as you can see, these are complementary regions, so they have bonded to each other. And this is three-dimensional structure or the actual structure of the transfer RNA. And this is a symbolic way to represent, as I have just drawn the diagram of a transfer RNA. So this is how you are going to draw a transfer RNA. You can draw it in any way. For example, a simple rectangle, and at one end you are going to show its, say, amino acid GLY, and then at the other end simply draw three lines and it will be an anticodon, and in case of glycine it will be CCA, or it can be CCU or CCG and CCC. So triple C is also a codon, anticodon for glycine. So for the most part, in describing translation or attachment of transfer RNA, so we usually draw it in this symbolic way, because this is a very complicated diagram. You are not supposed to memorize all this. You can draw this particular diagram. So this is how we represent transfer RNA. A 2D diagram, this is a simple 2D diagram, this is a 3D model, three-dimensional, and this is a symbolic way, the third one. As I just told you that transfer RNA molecules, they are interpreters, translators. They translate the messenger RNA code into amino acids. If you remember when we were discussing the transcription, so DNA carries information for all the RNAs found in the cell, whether it is a messenger RNA or transfer RNA or ribosomal RNA. So transfer RNAs are also transcribed from DNA. The difference between eukaryote and prokaryote is that in prokaryotes it's a single cell, so in that particular cell transfer RNAs are synthesized as DNA resides in the cytoplasm. But in eukaryotic cells, tRNA, the transfer RNA, is made in the nucleus and then it travels from the nucleus to the cytoplasm. So here is the tRNA, it will be synthesized in the nucleus and it will come or move from the nucleus into the cytoplasm where it carries out the interpreter function or the translation function. Now, transfer RNA is a kind of molecule which is used again and again, just like we have seen the example of a glycine transfer RNA. So it will bring a glycine into a growing polypeptide chain, leave it there, and this particular transfer RNA will leave the translation machinery, fetch or carry another glycine and then come back. So they are used again and again. Now, the basic function of a transfer RNA, what it does, that it picks up its designated amino acid, correct amino acids, and as I just told you, that each transfer RNA carry a specific amino acid. If it doesn't, then that will create problems, because the amino acid must match the anticodon which it is carrying. It must be according to the genetic code. So it picks up the designated amino acid. It brings that amino acid to the translation machinery. We are saying that depositing this cargo, that is the amino acid, into the polypeptide chain at the ribosome, and then leaves the ribosome and picks up another amino acid of the same kind, that is glycine or histidine or proline, whatever the case might be. So this is its function: picking up amino acid, bringing it to the translation machinery, adding it to the transfer RNA doesn't add it, but it is added to the growing polypeptide chain. So the transfer RNA becomes free when its amino acid is added to the polypeptide chain. So what it does, that it leaves and carries the same amino acid again if it is required in that particular polypeptide chain. So this was a basic introduction to the translation process or the components, and then we simply discussed in a very basic way the transfer RNA. In the next video, we are going to discuss, inshallah, the charging of the transfer RNA, how it carries its designated or specific amino acids. So we shall discuss that, inshallah, in the next video. Thanks for watching and see you in the next video.